So for methodology, we have three parts, which are culturing, process, counting, and also harvesting. So before doing all of these process, our group members had a discussion to prepare the data sheets and also the daily routine work schedule. So for the first part is the culturing process. In this process, the main thing that have to be highlighted is to sterilize the tools, all the tools that we're going to use, and also clean the hands of the person in charge to prevent contamination. So here are the methods. The next part is the counting process. So in this counting process, we'll have to prepare the culture, and temperature and light intensity in the inoculation have to be recorded, and also pictures were took to see the color changes of the culture. Here is the steps, and also we have to make sure that the flask is tightly covered with a cotton plug and also the aluminum foil. All the steps were repeated for every replicate. So proceed with the cell counting progress. Here, every person in charge in counting will have to use the very same method. Firstly, the very first thing to do is to swirl the flask so that the algae will not stick to the glass flask. Okay, we only have to select 3 small squares in the one big square and count the average in each of the squares. So next is the calculation for density. Here we use both hemacytometer and also Sedgwick rafter, so we have to count for both of them. The first one here is the calculation of cell densities for hemacytometer, and this is for the Sedgwick rafter. Okay, this is the calculation that we have to do for every cell counting. This is the formula for growth rate. This one is for the division time. And next is the harvesting method. The first thing to be done in the harvesting method is to pre-combust. So here we pre-combust 4 filter peppers, which are for Warner R1, Warner Replicate 2, F2R1 and F2R2. Next is the dry weight determination. Here we weigh every filter peppers to the 4 decimal value so that we get the accurate weight of the filter peppers. These steps are repeated for 3 days.
Okay, for the result, after calculating the phytoplankton on hemacytometer, we calculate the cell density using formula given, and here is the result of our density for the cell per milliliter squared against days that we count the phytoplankton. So you can see here for 1 and 2, the exponential start at day 1st to the 2nd day. Here you can see, and decreasing from day 2 to 3, and expectedly increase again at the day 3 and 4 here. And we are looking at Walner 1 with the orange color. There's slightly decrease from day 1 to 2, and moving on, increase from day 2 to 3. It's a bit different compared to Walner 1, even though it is the same medium. And now for F21 with the clear color, it's decreasing from 2 to 3 and slightly increase, which is the exponential phase, from day 2 to day 4. And expectedly for F2 replicate 2, it doesn't show the same towards the F21, even though it is the same medium. Okay, now we're going to continue with the division time. So how to get the division time is you have to divide 0.6931 with the growth rate that you have calculated in the previous slide, which is here. Through the cell density, you have to calculate the growth rate of the phytoplankton. Then only you can continue with the division time. As you can see here, the graph, all of the medium shows decreasing towards day 5 and 6 and rise again from day 6 to day 7. Okay, now we are looking at the biomass of the phytoplankton after we harvest them through the pre-combust, and we take the reading every day until the 4th day, which is supposed we have to take until constant, but we only looking to 4 days. So what we get here is the biomass against medium. So how to get the biomass against medium? First, you have to dry weight divide with the volume that you filtered. In this case, we are using 15 ml, so dry weight of the medium, you divide with 15 and you'll get the biomass here. So what you have to do is to construct the bar graph as shown right here. So only then you can get the biomass after you are going through the pre-combust and you wet the filter paper every day. So that's all for results.
Okay, now I want to talk about the growth rate and the division time for each medium. Okay, now you can see from this is the growth rate, division time and the phase. Why the growth rate is the proportion increase in cell number per unit time. Why division time is we can calculate it from the growth rate, which is the time for the cell to divide, but the unit is in days. This is the phases where the growth rate can be calculated. While for the 101, 102, the phases is exponential, while for the F2, F2 is stationary. We can see from this bar graph, the growth rate for the 102 is the highest, while for the F21 is the lowest. Okay, this is the graph of density of cells. Cell densities again this for each medium. Okay, first you can see from the walnut one, from day 1 to day 3, the cells start to increase. The graph showed increases. Well, this is because day 1 to day 3 is exponential phase. We can see from the color changes from 1 to 1, the 1, 2, 3, the color is still green, but after day 3, the color starts to changes, become pale color or to white color. For the reason factor that we can say is the light intensity is the lowest for day 3 other than other day. The 3 show the lowest light intensity, while for the 1 and 2, from day 1 to day 4 it show the increases because also the exponential phase for the 1 and 2. We can see for the color changes for 1 and 2, from day 1 to day 3 the color still in green color, but the color start to become the color start to changes light to light color or light green color or white, but increase the color changes back to the D5 to D7, which become D5 still become green back and become after that D6 and D7 they start to become light color light color of green. Okay, for F2 F2 sorry, for the light's intensity for 1 to 2, we can see it's still in the range of like good highest light intensity, which is above for 1000. Okay, for F21 we can see from D1 to D4, this is the stationary phases for F21. Okay, we can see from the D1 to D4 the color is still green, but after the D4 the color start to changes to white color, which so you can see from D7 actually the cells is in the death phase. Okay, see for the light intensity is still good for the F21. Okay, for F22 we can see that from day 1 to day 5 is the stationary for F22. For the color changes of F22 that we can relate is from day 1 to day 3, the color is still in green color, but you can see at the day 4 the graph is slightly down or decreases, because from the color changes it also show that the color is become white or something happened for the color changes, and back to green color on the fifth day to 7. As we can see the light intensity is high. Okay, for the light intensity for day 4 is not recorded by the member, so cannot be relate for the day 4 light intensity.
Okay, for our microalgae nanochlorosis, they prefer F2 medium compared to Walnuts medium. As we can see from the graph, if they are cultured in the Walnuts medium, they may face the sudden drop of cell density like the one I've showed in the graph. And there are two types of media that we use to culture this nanochlorosis, which is the F2 medium and the Walnuts medium. What makes these two medium different is the composition that made the medium. For example, in the walnuts medium, they are made up of nutrient, trace element, and vitamin solution, while in F2 medium, they are made up of uh, nutrient solution, vitamin solution, and sterilized seawater. Sufficient nutrient for growth is very essential and are very needed for the growth of of these nanochloropsis. And nitrate is one of the most important nutrient for their growth. So to compare these two medium, the nitrate solution vary for each medium. F2 medium contain a higher amount of nitrate and phosphate solution compared to walnuts medium. F2 medium contain 100 milligram per liter of nitrate and phosphate solution, while in walnuts medium, they contain only 75 milligram per liter of nitrate solution. Nitrate solution contain nitrogen. Nitrogen is very important of cellular protein and chlorophyll molecule that are required for the growth of the microalgae. Source and concentration of nitrogen have caused an important changes in the growth character and biochemical composition of the nanochloropsis. If they are cultured in the medium with low nitrogen or low nitrogen limitation,
like in the walnuts, it will result in the increase of the lipid content. To know the lipid content, we can is based on their dry cell weight or their biomass. For some reason, the increase in the lipid cell are also maybe because of they are in a stress state. Like we culture them, they are not in their initial or not in their initial condition. So which result in their decreasing or reducing in their cell density here, because there is less or limited content of nitrogen in the medium.
For group 4, we harvest the medium sample on the day 7. So harvest is the process of separate the algae from the medium culture. For this experiment, we harvest the sample on the day 7 because we want to get the biomass of algae before the algae undergoes the dead phase. So from this harvesting process, we can calculate and observe the biomass of algae. So what is biomass? Biomass is total quantity or weight of organism in the given volumes. Others meaning biomass is the organic matters used as fuel, especially in the power station for generation of electricity. In this experiment, algae was used to form the energy production like biofuels. So based on our data, we can conclude that F2 medium has highest biomass compared to walnut medium. This is because F2 medium has nitrogen that adds as supplement to the nanochlorobsis cell and make the 30% oil in their dry weight contain. This increased the number of biomass of the nanochlorobsis in the F2 medium. For the precautions, we need to make sure that all apparatus and hand was sterilized before doing the experiment, especially what's on the culture. So before we want to do the culture, we make sure that all apparatus and our hands are sterilized with the ethanol. Second, the way of preparing the slide must be standardized with all of team members. For example, we put the cover slip first on the hematocytometer slide before we put the samples. Third, keep the flask on the medium culture at the same position. Flask of the medium culture must be at same position on the first day until the end of experiment. Number 4, shake well the flask before take the sample to make sure the sample are mixed well. So before we want to take the sample for the calculate the density, we need to shake well the sample for the sample mix well. And lastly, fix the decimal point of the weight of filter paper.
Now we move to the conclusions. For nanochlorobsis, it favors F2 medium more than the walnut medium. It is because F2 medium provide nitrogen that help the growth of nanochlorobsis. But still, in the walnut medium, nanochlorobsis still can grow, but the growth much slower than nanochlorobsis in the F2 medium. So basically, this experiment cannot fully accept it because actually the F2 medium contaminated at day 4. So lastly, taking precautions are very important to this experiment. The precaution that should we take already stated in the discussion. Thank you.